today is the second section of chapter 5 on homeostasis and transport, which is all about active transport. Now, in the last section, when I described passive transport, I described it as being passive because it does not require energy. And so active transport is going to be the opposite of that. It's going to require energy because you're moving a substance up the concentration gradient from low concentration to high. All of the passive transport processes described: diffusion, osmosis, facilitated diffusion, and ion channels are all the movement of molecules down their concentration gradient from high to low. This is going to be the opposite, which is why it requires energy. And again, if you think about the analogy that I used of rolling a ball down a hill, if you're at the top of the hill and you let go of a ball, it goes down the hill by itself, high to low, doesn't require energy. But if you're at the bottom of the hill and you want that ball to go up to the top of the hill, it requires an exertion or input of energy from you. It doesn't do it by itself. So this is called active transport. There's going to be 3 major processes I'll go through. One of them is called the sodium-potassium pump. And there'll be a reason why it's called pump. And then I'm going to talk about exocytosis and endocytosis, which I know sound super complicated, but again, break down the word, understand what the parts of the words mean. It's not so scary. So carrier proteins not only help in facilitated diffusion, but they can also help with active transport as well. When carrier proteins are using active transport, they are referred to as pumps because they're moving substances against the gradient. The process of membrane pump is going to be similar to facilitated diffusion, with the protein changing shape. So as soon as the molecule binds to the protein changes shape and opens up to the other side of the membrane, and the substance is moved inward or outward, depending on which direction you're going. So the most common pump that is studied in biology, at least at the high school level, is called the sodium-potassium pump. Now, I'm just going to use this animation to describe what happens. We're going to go in steps, okay? So the first thing that happens is 3 sodium ions go inside the protein, which you can see happening, the 3 round pink ones there. When that happens, a phosphate group is taken off of an ATP, and the phosphate group is attached to the protein. So you see, as soon as the 3 are attached, the protein changes shape, and you've got that little P in the corner here. And yeah, so right in the beginning, you can see it's kind of cut off because I just have it on a loop, but you can see the ATP kind of dropping off. So as soon as the 3 sodium ions go in, then you've got the ATP, which releases the phosphate. The phosphate changes the shape of the protein. Now, after that, the 3 sodium ions are released to the outside of the cell. So it's important to understand this is inside, this is outside. And notice that the left side is matching the shape for sodium, and the right side matches the shape for potassium. So as soon as the protein opens towards the outside, the 3 sodium are released and 2 potassium ions enter the protein. Now, as soon as that happens, you see that the protein changes shape and the phosphate is released. The max speed of this pump is basically 150 cycles per second. So imagine seeing what's happening here. That happens 150 times per second. So every second you're pumping out 450 sodium and 300 potassium in. It should be pretty simple because we already hopefully understand how facilitated diffusion works, which is basically the same thing, but without the ATP. That's the only difference between the sodium potassium pump and the facilitated diffusion I talked about last week. So the only difference is here you need a phosphate.
This section of a cell membrane contains a carrier protein called a sodium-potassium pump. This protein...
You can hear that, right?
Yes.
Okay. So then that does work. All right.
This section of a cell membrane contains a carrier protein called a sodium-potassium pump. This protein moves sodium ions and potassium ions against their concentration gradient. Such movement requires the cell to use energy and is therefore a type of active transport.
Okay, so first thing I want you to do is look at the concentrations on either side. So in this case, you have more red squares on the inside. So the red squares are going to be potassium, and you have more purple triangles on the outside. That's going to be sodium. So you have a high concentration of sodium outside and a low concentration of sodium inside, and yet you're going to pump sodium out from low to high. You have a high concentration of potassium inside and a low concentration of potassium outside, and yet you're going to pump potassium in. That's active transport.
The energy needed to operate a sodium-potassium pump comes from a molecule called ATP. Notice that the sodium-potassium pump moves sodium ions and potassium ions in opposite directions. The pump moves sodium ions out of the cell and potassium ions into the cell.
Okay.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an
There's
ATP
ATP.
provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure.
Oh, I'm sorry. That's my fault. Never mind. I went through it anyway. Okay, so the purpose. You might ask yourself, what is the purpose of actually needing to use energy? Oh no, there we go. What is the purpose of using energy to pump sodium into an area which already has a higher concentration and potassium in for the same reason? The purpose behind this is to create an electrical gradient. Now, we already hopefully understand what gradient means. It's having 2 different concentrations of something on either side of a membrane. So if this is our cell membrane, we're pumping out, let's say per second, we're pumping out 450 sodium and we're only pumping in 300 potassium. Both of these ions have a charge of +1. So every second, you are creating a gradient of a difference of 150, a charge of 150. Now, why would we want to create an electrical gradient, you might ask? It's extremely important for the transmission of nerve impulses. I don't know if you guys, you're probably too young to have seen the movies The Matrix. or maybe you have, like watching them as like, oh, these are old movies. But part of the idea of the Matrix is that basically these big giant robot things have taken over the world, and they are growing human beings as a source of electricity because our brain communicates with our body through electrical impulses. And so I don't know if you studied electricity in physics yet, but electricity flows due to current, and that current is due to a difference in potential, meaning the let's say like the affinity for charge at either end of a circuit is different. So it's a gradient of sorts. And so that's the point of the sodium potassium pump. Next year, when we look at impulses and how they work, you're going to see the sodium potassium pump in action. Basically, when an impulse goes through a nerve cell, it completely puts ions on the wrong side of the membranes, and the sodium potassium pump is the one that puts everything back where it's supposed to be. So outside, more sodium is being pumped out than potassium is being pumped in. So there's a more positive charge outside of the cell than inside the cell. This is what allows the impulse to go through a nerve cell. So it's important for nerve impulses. All right, and for the last two, endocytosis, exocytosis. Again, they sound like super scary words, but you just break it down. Endocytosis, exocytosis. Cis process, if I can spell, cyto, cell, endo, inside, exo, outside. So some kind of process involving the cell and bringing things inside. And then exocytosis is a cell process that exports things outside. You have actually already seen endocytosis and exocytosis in action. You just didn't know what they were called. And I actually might have mentioned it, I'm not sure. But anyway, some molecules are too large to pass through the cell membrane or the carrier proteins. Or sometimes your cell wants to bring in large amounts of a substance that are not going to either go through the lipid bilayer, or it's too much to go through the carrier proteins. So these are used to transport either large molecules or large amounts of small molecules. Again, endo means in, exo means out. Now I was trying to think in my brain while I was talking, when have you seen this? When I talked about the endoplasmic reticulum, the ribosomes, rough endoplasmic reticulum, and Golgi body, how they all cooperate to export proteins. That process of the vesicle from the Golgi body going to the cell membrane, fusing with the cell membrane, and releasing the proteins to the outside of the cell, that was exocytosis. These two processes are going to be exactly the same thing, only opposite. And that kind of sounds weird when I say it that way. Exactly the same thing, only opposite. It's the same process, they just go in opposite directions, okay? So endocytosis, again, with the phonetic spelling, endocytosis. That's how you pronounce it. This is when a cell can ingest or bring in external fluid, macromolecules, or large particles. And you can see how it works. And this is why the lipid bilayer is so important, because not only is your plasma membrane made of the lipid bilayer, but everything that travels into your cell, out of your cell, around in your cell is done through vesicles. And look at the structure of the vesicle. It's literally a hollow ball that's made of lipid bilayer. This is the reason why you can very easily bring things in and out of the cell, because you're just making it from these lipid bilayers. So if you've got a large amount of stuff, the cell membrane starts to fold in, all of those substances go inward, eventually it pinches off and you get a vesicle. That's it. Now, if this is food, then you're going to have your lysosomes, which are also, here's your lysosomes, and I'll do my lipid bilayer. Those are the tails. And inside is enzyme. So then your lysosome will fuse with the food vesicle, and it will break down, the enzymes will break down the food inside the cell. It could also be a way of protecting your body against, what do you call it? Pathogens. So here we go. This is a diagram of a process called pinocytosis. And this is an actual photograph. So I thought that was pretty cool to add. So under the microscope, you see these pockets being formed. And that's because the cell is in the process of ingesting solutes or liquids. So there's two types of endocytosis depending on what you're bringing in. You don't really see pinocytosis being referred to very much. Like, I think besides this chapter, there's no other situation where it's mentioned. But just to distinguish it from the other kind of endocytosis, which does get talked about a lot in other chapters, which is phagocytosis. Phagocytosis is moving large particles, but for the most part, it's whole cells. This is extremely important for your immune system. What you have here is what's called a macrophage. Macrophage, which is a type of white blood cell, and you see that it shares the same root with PHAG, phag, and macro meaning large. So a macrophage is a white blood cell that's going to use the process of phagocytosis, and that's why it's called a macrophage. So here you've got a bacteria, oh no, and the white blood cell is going to use phagocytosis, which is the specific kind of endocytosis, to engulf it. And then once inside, the macrophage is filled with enzymes again, and it will break down the bacteria. And finally, exocytosis. This is just the reverse. This is the one that I talked about in chapter 4, which feels like a semester ago, even though we're only on chapter 5. It's so weird. When you go from the ribosomes, where the proteins are made on the ER, they travel through the ER to the Golgi body, and then they get surrounded by a vesicle membrane, goes to the cell membrane, fuses with the cell membrane, and then the proteins are released to the outside. And so again, this is, we call it micrograph, which is a microscopic picture. And you can see the bilayer here. And the vesicle is in the process of fusing and releasing the contents of the cell. I don't know why it's so pixely, but anyway, and the diagram, same thing, you've seen that before. So anything that's exported out of the cell is going to be exported this way.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane. They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle. The vesicle moves to a stack of membranes called the Golgi apparatus. Their membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane. This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.